All right, today we're going to dive into a really fascinating condition called amyloidosis. It's basically a story about how proteins, the building blocks of our bodies, can sometimes go completely rogue. We're going to break down exactly how that happens, step by step. So here's our game plan. First, we'll get a handle on what amyloidosis actually is. Then, we'll meet the three main villains of the story. After that, we'll look at how doctors figure it out. And finally, we'll explore the really clever strategies used to fight it. Okay, let's start at the very beginning. What in the world is amyloidosis? Well, the name itself actually gives us a huge clue. It comes from two Greek words, amyloid, which means protein, and osis, which means too much of something. So put them together and you get excessive protein. At its very core, that's what we're dealing with. But here's where it gets really interesting. It's not just about having too much protein. It's about that protein being shaped all wrong. Think of proteins like tiny, complicated origami. When they're folded correctly, they do their job perfectly. But in amyloidosis, something goes wrong in the folding process. They become these useless, sticky messes that the body just can't clear out. So they clump together and build up in our organs, causing them to malfunction and eventually fail. So, and this is a crucial point, amyloidosis isn't just one single disease. It's more of an umbrella term. Which type you have depends entirely on which specific protein is misbehaving. So let's meet the three main culprits. And that leads to the big question, right? Why do these proteins start misfolding in the first place? Well, there isn't one simple answer. Instead, there are three totally distinct pathways that can lead to this protein pileup. This table is a fantastic little cheat sheet. As you can see, we've got AL amyloidosis, which comes from a problem with plasma cells. Then there's AA, which is triggered by chronic inflammation. And finally, ATTR, which is either genetic or just a consequence of getting older. Okay, first up is AL amyloidosis. This is the most common type, and the problem here starts deep inside our bone marrow, with the plasma cells that are supposed to help our immune system. You can really see the domino effect here. It starts with faulty plasma cells, maybe from a condition like multiple myeloma. These malfunctioning cells start pumping out way too many unstable protein bits called light chains. These chains misfold, stick together, and start clogging up vital organs, especially the heart and kidneys. All right, let's move on to our second type, AA amyloidosis. Now, this one is called secondary for a really important reason. It doesn't just happen on its own. It's a reaction to another problem that's been going on for a long time, chronic inflammation. So when the body is dealing with long-term inflammation from conditions like these, the liver basically goes into overdrive. It starts producing this protein called serum amyloid A, or SAA, and if there's too much SAA for too long, it can become unstable, misfold, and build up in organs. This time, the kidneys are usually the main target. And finally, that brings us to our third main type, ATTR amyloidosis. This one is all about a protein made in the liver called transthyretin. We'll just call it TTR. Its day job is to carry thyroid hormone and vitamin A through the body. Now, this type is actually a two-for-one deal. First, there's the hereditary form, HATTR, which is caused by a faulty gene you're born with. Then there's the wild type, or WATTR, which happens when the normal, healthy protein just gets unstable as we age, usually after 65. The hereditary form tends to affect the heart and nerves, while the age-related one mainly goes for the heart. Okay, so we've met our three main culprits. But how do doctors play detective to figure out not just if it's amyloidosis, but exactly which type is causing all the trouble? Well, the investigation starts with looking for clues. Sometimes there are physical signs like an enlarged tongue, but the lab tests are really crucial. Doctors will hunt for those abnormal light chains in the blood and urine to spot AL, they'll look for genetic mutations to find ATTR, and they'll check for high inflammation markers that point to AA. But the definitive proof, the absolute smoking gun, is visual. They take a tiny tissue sample, a biopsy, from an affected organ. They stain it with a special dye called Congo red, and then they look at it under a polarized light microscope. If it's amyloid, the deposits light up with this unmistakable, almost beautiful apple green glow. That's the moment you know for sure. So once the diagnosis is confirmed, it's time to launch a counterattack. And this part is absolutely key. The treatment has to be perfectly tailored to the specific type of amyloidosis they found. And that really is the core principle of treatment. You can't just throw one drug at all three types. The logic is so direct and honestly kind of elegant. For AL, you have to stop the source. For AA, you stop the trigger. And for ATR, you have to stabilize the wobbly protein itself. So for AL amyloidosis, because the problem is those rogue plasma cells, the treatment looks a lot like what you'd see for cancer. The goal is to shut down the factory that's producing all those bad light chains, using chemotherapy or even a bone marrow transplant to completely reboot the system. With AA, the logic is beautifully simple. You just go after the root cause. If you can treat the underlying chronic inflammation, the liver gets the message and chills out. It stops overproducing that SAA protein, and the whole damaging process just grinds to a halt. Now, for ATTR, the approach is completely different yet again. We don't need to destroy anything. The whole goal here is to stabilize that wobbly TTR protein before it gets a chance to break apart and cause trouble. And this is just a really elegant solution. A drug like tafamidis basically acts like a piece of molecular tape or glue. It physically latches onto the TTR protein, holding its structure together so it can't fall apart and create those dangerous clumps. So if there is one thing you take away from all of this, just remember the number 3. 3 main types of amyloidosis, each with its own unique cause, and each requiring its own unique, highly targeted solution. You know, whether it's from faulty cells, chronic inflammation, or just the process of aging. Amyloidosis is such a powerful reminder of how a tiny, tiny error in a protein's shape can have massive consequences for the whole body. It really makes you wonder, as our science gets better and better, what does the future hold? Could we one day treat these diseases at their most fundamental level before they even start?